Okay. What we'll talk about today is going to start with Protozoa. Okay. Should I write a little bit bigger letters, or you're okay? No, we good? Okay. So Protozoa, by definition, are unicellular, cellular. Okay. So can be divided into four relatively broad categories. And please understand that these categories are not essentially taxonomical ones, that those are not phyla or classes or general orders. This classification is loosely based on the mode of motion that particular protozoa utilize. So we've had discussed before Sarcotina. Okay, we've discussed it before, Sarcotina, which use amoeboid motion, amoebic motion, forming pseudopod and crawling around. Mastigophora that use flagella for moving around. Celiophora that obviously used cilia for moving around. And finally, Apicomplexa, which are non-moving microorganisms. Non-moving microorganisms. Let me check. Angelina, can you hear me?
Yeah, I can hear you now.
Okay, awesome. Awesome. So non-moving microorganisms, and therefore Apicomplexa are obligate parasites. So I wanted to mention a couple more features of the typical protozoan cell. First, typical protozoan cell is kind of two layers of a cytoplasm. The outer ectoplasm is responsible for moving, feeding, the activities that involve the interaction with the outside environment, while endoplasm contains all the internal structures of cell, such as mitochondria, apical complex, contractile vacuole, and so on and so forth. Another important feature of protozoan cells is their ability, for most of them, to form so-called cysts. So the living and feeding form of a protozoa is called a trophozoite. So tropho means feeding, zoa means living, so feeding, living, reproducing form. Under the harsh environmental conditions, though, protozoa can form cysts. Cysts cannot replicate. Cysts cannot feed. Cysts are basically a sleeping cell. But when environmental conditions become good, cysts can become trophozoite. It's important to understand that both trophozoites and cysts can lead to the infection. Trophozoite directly leads, you can consume a protozoa in the form of trophozoite and will cause the disease, or you can consume a cyst, and cyst in your will become trophozoite and cause some kind of a problem. Does that make sense? Good? Okay, having said that, we're going to start our conversation, the discussion of our first group of protozoa, which will be amoeba. So in that group, the first pretty large category, I'm trying to find all my different markers. First category will be amoeba that cause granulomatous meningoencephalitis. So in this group, there are two species, or two, even I would put them as a genera that can do that. Those are Acanthamoeba castellani and other relatives in that group, and Balamuthia mandrillaris and other relatives of that amoeba. Acanthamoeba and Balamuthia can be transferred by a broken skin, So these are soil microorganisms. So when people walk around, work on the soil, work on the, in the garden, or kids are playing around, and they are on their knees and they scratch the knees, this is how this amoeba can enter the circulation. These amoeba are common for tropical regions, so you're not going to find those in the United States. So they enter into the circulation and end up in the brain, and they cause inflammation of the meninges and of the brain, meningoencephalitis, with the formation of granuloma. So typical signs of meningoencephalitis that include confusion, headaches, fever. Now, this disease is treatable, and what is very important, this disease is non-communicable. So this infection cannot be transmitted from a person to another person. The second group of amoebae that I wanted to address was, let's call it gastrointestinal disease. So that will be Entamoeba histolytica. Now, Entamoeba histolytica is transmitted by fecal-oral route, most frequently due to the contamination of water or maybe food. And it causes dysentery, colitis, and intestinal perforation. So due to the intestinal perforation, this microorganism may end up in circulation and migrate to different organs. For example, livers and lungs. And in the liver and the lungs, it can cause so-called amoeboma. These so-called extra-intestinal amoebomas, characteristic for liver and lungs, look like a tubercle, a granuloma. Basically, the cells that are infected with amoebae cannot be cleared by the immune system, so the immune system forms a layer of fibrin around the infected focus in order to isolate that infected focus from the rest of the body. So far, does that make sense? Good. Any questions? Okay, if no questions here, and feel free to ask the questions, because some of the stories that we're gonna work talk about here. Pretty interesting. So we're going to talk about now we're going to talk about perhaps the most important protozoan pathogen in the world, which is malaria. So malaria is caused by the species of Plasmodium. So this entire genus, Plasmodium, can cause malaria. Many species can cause malaria in humans. Many species can cause malaria in animals. Plasmodium is apicomplexa, which means that this microorganism cannot move, and therefore it is obligate parasite. So let's start with the fact that it's obligate parasite. And it is transmitted directly via biological vector. So it's transmitted directly by the mosquitoes from person to person, from one infected person to infected person. You may have already looked in the lecture notes and saw the life cycle, which is very detailed. Now, I do not have an intention of discussing that life cycle in that amount of details. I want to focus on certain steps. For instance, in that life cycle, you may notice different names for different forms of Plasmodium. Sporozoid that infects liver cells, murozoid that infects red blood cells. I do not require you to know those life forms. I want you to have, though, pretty decent understanding of what's going on during the human infection with Plasmodium. So first, we have to start, of course, with a mosquito bite. Okay, so mosquito takes a blood meal and transfers the parasite into the circulation. And parasite immediately, I'm going to call Plasmodium PL. So Plasmodium ends up in the liver. Okay. After the replication in the liver cells, Plasmodium is released from the liver and it infects red blood cells. It's kind of interesting. As far as I know, to my knowledge, Plasmodium and other members of, that genus and related genera that can cause this malaria-like illness, those are the only parasites that actually infect red blood cells. So what can happen in the red blood cell, and why does Plasmodium infect it? Plasmodium needs basically the protein component of hemoglobin for its growth and reproduction. Now, it grows and reproduces inside of the red blood cell, and it leaves red blood cell every so often, producing new plasmodia that can infect red blood cell. So it's kind of, a cycle. Does that make sense? So microorganism gets out of the red blood cell, bursting it up, infects more red blood cells and more red blood cells and more red blood cells. We're going to get back to the symptoms a bit later. Now, another possible outcome of the infection of the red blood cell is the production of gametes, male and female. Now, the proper term for gametes are gametocytes, which I quite frankly don't care much about, okay? The point is that there are sexual cells produced. The sexual cells, the gametes, are getting in the mosquito when mosquito takes a blood meal, so another mosquito bite. And in the mosquitoes, male and female gametes fuse, forming a zygote. In the mosquito. Zygote in the mosquito develops in the adult plasmodium, again, in the mosquito, and the cycle starts over. So one more time. Mosquito takes a blood meal, takes a bite. Plasmodium enters the circulation and migrates to the liver, where it infects liver, which, by the way, leads to hepatomegaly, enlarged liver. Then Plasmodium infects red blood cells. It gets out of the liver, infects red blood cells. Okay? So it gets released from red blood cells every so often, infects more red blood cells. Those red blood cells are being recycled in the spleen. Okay, and that recycling of red blood cells in the spleen leads to splenomegaly, increased recycling of red blood cells in the spleen. Also, red blood cells produce gametes, which are taken up by a mosquito during a second blood meal. Gametes fuse, forming a zygote. Zygote develops in the adult plasmodium in the mosquito, and the cycle starts over. Does that make sense? Awesome. So here in this cycle, I want to highlight a couple of new terms for you. One is a definitive host. A mosquito here is the definitive host. Why we call it a definitive host? Because that's where the sexual stage happens. That's where gametes fuse to form a zygote. The human, I don't have a human here, but you get the gist. Human, on the other hand, is intermediate host. Okay? And we're going to use the term definitive and intermediate host a lot today. So far, we good? So what are the symptoms? So primary infection results in a fever and back pain. Then this microorganism, in kids particularly, it may travel to the brain and cause so-called cerebral malaria, which is very dangerous. The patient may slip into coma and never recover from it. In fact, 70% of people who die from malaria are children, age from 6 months to about 5 years. Because that's the time when maternal antibodies wane, they kind of wear off, but they're more susceptible to that disease. Now, what the disease looks like in the adult. So after the initial period of the back pain and fever, the person will experience recurrent bouts of fever, chills, profuse sweating, and shakes. Okay? Now, how frequent those bouts occur? For different species, they happen at different times. Essentially, the species that infect a particular patient will determine the frequency of malaria symptoms. I don't remember myself which species have what frequency. So, like, I think Plasmodium malariae has like every 2 weeks. Plasmodium falciparum is every 3 days. But basically, every time Plasmodium ruptures out of the red blood cells, that's when those shakes and chills occur. Does that make sense? So it's species that determine the symptoms of the disease. There is no vaccine. I'm going to put it here. But this disease is treatable. The infamous hydroxychloroquine, along with drugs like mefloquine, quinine, quinidine, artemisinin, all those drugs can basically cure malaria. However, according to the CDC guidelines, blood donation guidelines, basically, once you had malaria, you are considered to be positive for life. Does that make sense? Okay, good. Any questions? The Plasmodium? No? Okay. How many, does anyone have a cat? Okay, I can only see Grace, so I presume that Grace does have a cat, and I don't know about anybody else, because I don't see pictures. I don't see images. Okay, I'm going to ruin the cat for you now, because we're going to talk about Toxoplasma gondii. So Toxoplasma gondii is another example of apicomplex and parasite. And it is a pretty interesting microorganism. We're going to talk about the life cycle first, and we're going to talk about two life cycles. So Toxoplasma is acquired by virtually all species via fecal oral. Well, okay, let's say with the foot. Let's not talk fecal oral, okay? Let's say with the foot. And the first route that I want to describe here is, so we have rodents that are infected with Toxoplasma, and they shed cysts in their feces. Now, I'm not going to put, because it's always either feces or food, so the fecal of rodents ends up, contaminating stuff, and then this fecal matter is consumed by cattle or pigs. I mean, of course, sheep are also included. Herbivores. And then those herbivores are consumed by humans. Now, it's important to understand that the consumption here, you have to consume undercooked beef or undercooked pork. Does that make sense? So, like, if you cook it properly, you're fine. But if you like your steak rare, then there's a pretty good chance that you're going to get Toxoplasma. In fact, in the countries where they, well, literally like they steak rare, I'm talking Brazil and France, the prevalence of toxoplasmosis in the general population reaches about 80%. In the United States, it's about 22-23%. So what it's going to do to you if you are immunocompetent individual? Nothing. Literally nothing. Like, you can be infected with Toxoplasma and you will have no symptoms whatsoever. What it can do to you if you are immunocompromised. It's going to cause encephalitis, and in fact, encephalitis is a Toxoplasma infection of the is a really common infection in patients with AIDS. Now, why I told you that I'm going to ruin your cat for you? Because, as you can imagine, rodents may be consumed, not only, well, not rodents, but rodent feces can be consumed by cattle and pigs, but rodents themselves, such as mice, can be consumed by cats. Now, this is an extraordinary transmission route here. It's one of the very few examples where microorganism modifies the behavior of the host in order to facilitate transmission. Turns out that mice that are infected with Toxoplasma, they stop fearing cat's urine. Mice can smell the urine, the smell of the cat's urine, pretty well, and they're going to stay away from a cat. When they're infected, they don't give a crap anymore, and it's much easier for cats to chase them and catch them, and basically imagine that Jerry is now completely nuts and just approaches Tom nonchalantly, and Tom basically, that's the end of the whole Tom and Jerry story. And then when cats poop, they poop the cysts of Toxoplasma, and cysts end up in the litter boxes, and people tend to the litter boxes, and that leads to human infection. This is what, and another problem, why it's a problem. So if you are immunocompetent and you have Toxoplasma infection, it's not a big deal, right? Or if your cat is completely domesticated, if there are no mice involved, you're not at risk here. But generally speaking, since Toxoplasma can be, so we get vertical transmission to fetus, okay? A pregnant woman that is infected with Toxoplasma can give it to fetus, and fetal toxoplasmosis is extremely severe. This is why pregnant women are advised to stay away from cats and litter boxes. Does that make sense? Good. Well, I hope I didn't ruin the. Go ahead.
The ammonia in the litter, like cat urine. That's why
Say it again?
I thought the ammonia in cat urine was like a big factor in like pregnant women, like why they shouldn't clean the litter box.
Oh, sorry. Your sound was going was not very clear in the beginning of the sentence. Have you heard about this?
No, but I've heard of like women not cleaning out the litter box because of the ammonia.
Toxoplasma can cause pneumonia, but the main concern, quite frankly, for pregnant women is this. Like, seriously, that is a big no when we're talking about cats and pregnant women. I mean, cats are okay, but litter boxes are no-no. Okay? Now, I mentioned that it's one of the, well, if not the most successful parasite in the world. Why? Estimates put the number of people that are infected at about half of the world population, which is a lot. Okay? Like, that's really far-reaching estimates, but still, we're talking about billions of people being infected with the Toxoplasma. Okay? That makes sense? Now, last protozoa that, if you don't mind, we're going to talk protozoa, then I'm going to ask a 5 minutes break, and then I'm going to go back and we're going to finish, wrap the whole thing up, okay? So last protozoa that we're going to discuss now is Leishmania donovani. Well, sorry, Leishmania. So Leishmania species, such as Leishmania donovani, Leishmania panamensis, or Leishmania brasiliensis, they all cause the disease called leishmaniasis. This microorganism is transmitted via biological vector, a scent fly. So when scent fly takes a bite, it leads to first the cutaneous leishmaniasis, that manifests as a lesion on the skin, and it's restricted to the skin with elevated edges. In immunocompetent individuals, most cases, this lesion can persist for weeks and may be healed eventually, okay? If the infection is, so more severe form of leishmaniasis is mucocutaneous, or Bispundia, when infection spreads to the mucous membranes, most frequently the mucous membranes of the oral cavity, and that may lead to the complete destruction of the soft palate that will require medical intervention. And the most severe is visceral leishmaniasis, which implies multi-organ involvement, hepatomegaly, splenomegaly, And if untreated, visceral leishmaniasis is uniformly lethal. Leishmania infection can be treated. There are drugs, but again, if you ever have been infected with leishmania, you cannot donate blood. So basically, treatment is not considered to be curative, sterilizing. Does that make sense? I'm going to pause the recording. So now we're going to talk about my second favorite microorganisms, helminths. So viruses, and basically viruses take my left ventricle and helminths take my right ventricle. So, helminths that cause human infections, and okay, let me clarify. We're going to talk specifically about the infections that occur inside of the body, like deep inside. There is a helminth infestation called myiasis. Yeah, I think it's myiasis. I'm not sure that I pronounce it correctly. So basically, it's when I think it's black flies that deposit their eggs into this. So eggs develop in the larva, and larvae burrow into the skin of a human, and they develop into an adult fly in the skin. I know it sounds pretty disturbing, but we're not going to talk about that type of infestation. We're going to focus on several helminthic infections that occur kind of, deep. But first, we need to figure out the classification of the helminths. So the first group of helminths is nematodes. Nematodes are roundworms. They do have body cavity. So they actually have a whole bunch of internal organs. And they have sexes. So they have males and females. Therefore, for reproduction, males and females both have to be present for a male to fertilize the eggs. Second group are trematodes. Trematodes are flat, non-segmented worms that have no body cavity, and they are hermaphrodites. Now, to make it clear, there is one little exception in this group, but for our intents and purposes, trematodes are hermaphrodites. And finally, the third group is cestodes, which are flat, segmented worms, also hermaphrodites. Now, you may know that helminthic infestations can be fairly easily treated. Let me see if I think that's better. Helminthic infestations can be fairly easily treated. Now, I want to highlight two major groups of anti-helminthic drugs here. Now, first group will be what I personally call bendazoles. You have some examples in the lecture notes, like mebendazole, albendazole, thiabendazole. So basically, if you see bendazole, you know what the drug is. And the mechanism is that bendazoles inhibit microtubule synthesis. As you can imagine, one of the major challenges in treating helminthic infestations are, first of all, the fact that helminths are eukaryotes, so you have to have something that affects specifically helminths but not humans, and the fact that you cannot target reproduction. The disease associated with the helmintic infestation is caused by an adult rather than the process of its replication. So you have to be able to immobilize, to paralyze, or to disintegrate an adult worm. And inhibition of microtubule synthesis actually leads to impaired cell division and eventually the death of an adult worm. Another drug that actually is so good that it deserved a Nobel Prize, and it received a Nobel Prize. Well, not the drug, but the people that discovered it. Ivermectin. So ivermectin is one of the main components, not one, okay, it's a main component of drugs that we use to prevent heartworm in dogs. In humans, it is exceptionally successful in treating various nematode infestations, which allowed countries in Africa to completely eliminate diseases such as river blindness or dracunculiasis. So ivermectin is quite fascinating. So ivermectin binds to the chloride channels, and it opens chloride channels on the worm cells, which leads to the chloride influx, which leads to hyperpolarization and hyperpolarization of worm cells leads to the paralysis of the worm. Okay, if it's a muscle cell and it doesn't work, that's paralysis. If it's a nerve cell and it doesn't work, it's paralysis as well. So far, we good? Okay. Any questions on this? Right, still. Let's tackle our first worm for today, which will be Technococcus granulosus. Technococcus granulosus is a cestode, segmented flatworm. And here's the life cycle. So in the life cycle of this worm, the adult is present in dogs. Okay? Now, adult worm sheds eggs. Okay? And eggs and feces are consumed by sheep or goat. Okay? And that's where these eggs develop into larvae. Now, what happens next? Sheep or goats get butchered, but we use meat, not the innards. And especially on like small farms or farms in the underprivileged communities, the insides of a sheep or a goat will not thrown away, but will be given to local kind of farm dogs as a food. So those dogs will basically consume larvae, and this larva will develop into the adult worm in the dogs. That make sense? So the main organ where Technococcus granulosus develop is liver. Okay? that make sense so far? Now, the best way to break down the cycle is to stop feeding dogs with the innards of butchered animals. Make sense? Because if you don't do it, then this is going to happen. Eggs in dog feces can end up on the human food. You know, just it's normal. Fecal contamination of food is normal. And eggs can permanently get in a human, leading to larvae being in the liver. Okay? And that will lead to so-called hydatid cysts in the liver. Cysts filled with the fluid where echinococcal larvae float. So far, does that make sense? Now, in this grand scheme of things, there are different roles that are assigned to different animals. So dogs will be definitive host. Sheep or goat will be intermediate host. And humans will be what is called accidental host. Because last time I checked, humans do not routinely get butchered and their livers not thrown to the dogs as a meat. Concern, along with damage to the liver and all associated problems, such as indigestion, fat intolerance, jaundice, and so on and so forth. If this hydatid cyst ruptures and the fluid from hydatid cyst float, gets into the rest of the organism, it causes anaphylactic shock, and a person dies of anaphylactic shock in a matter of minutes. This happened before when, those cysts accidentally ruptured, not during the surgery, but just, hit in the right hypochondriac quadrant, and that's it. That make sense? Any questions on this one? Okay, next one is a really fascinating helminth. It's a pretty large genus of Schistosoma. Schistosoma species are nematodes or flukes. And here's their life cycle for your enjoyment. So it all starts with the eggs in the environment. And then these eggs are picked up by snails, where they develop into larvae. Now, Schistosoma larva actually undergoes several transformations. All these stages have different names. You know them. In the end, this larva, the proper term for it is a cercariate, but I don't require you to know that. Then this larva end up floating freely in the water. So how can you get larva from the water into a human? You would say, well, you probably drink it. No. Turns out that adult worm in humans gets developed after larva penetrates unbroken skin. May sound pretty creepy, and it is. So imagine you are swimming in the water of some African or Southeast Asian lake, and you get out on the shore and you don't have a towel. So you decide that the water will dry out. When it dries out, this larva crawl into the hair follicles and penetrate from the hair follicles into the circulation. So they start with the skin capillaries and then eventually end up in the systemic circulation. This is why another name for schistosoma is a blood fluke. And from the systemic circulation, they end up in the, I'm going to highlight it here. So I'm going to go peri-intestinal and I don't know how, well, peri-intestinal capillaries. We're going to start with the peri-intestinal capillaries. And capillaries around urinary bladder. Does that make sense? The capillaries that surround intestine and capillaries that surround urinary bladder. So capillaries that surround intestine are the part of the portal system, you know, that drives the blood from the intestine to the heart, to the liver, I'm sorry, from the intestine to the liver, of course. So the infection of peri-intestinal capillaries may lead to portal hypertension. Portal hypertension, liver failure, hepatomegaly. The microorganism may spread systemically, causing neuroschistosomiasis, infection of mostly the spinal cord, and schistosomal infection of around the urinary bladder may lead to cystitis, and ultimately to the bladder cancer. It's a pretty well-known complication of chronic schistosomal infection. And of course, the microorganisms will shed the eggs in the environment. Does that make sense? So in this scheme of things, snails will be the intermediate host. And humans will be a definitive host. So obviously this is more of a long-term complications of schistosomiasis. So what does primary schistosomiasis look like? In the primary schistosomiasis, so you dried up on the shore of a lake, what can you see 2 weeks later? You will develop fever, so-called Karayama fever, and rash on the parts of the body that were covered in water. There were cases of people like standing in the lake and then drying up, and only their legs will be affected by that rash because that's where the larvae penetrate the skin. Does that make sense? So basically, if you're in Africa and you're swimming in the lake, if you get on the shore and not eaten by crocodiles, dry yourself with a towel to avoid schistosomiasis. We're good so far? Questions? No? Okay. Let's move on to another trematode, a microorganism called Fascial Hepatica. So as you can tell from the name, it has something to do with the liver. So life cycle is very similar to what we just discussed in regards to schistosoma. Eggs in the environment get picked up by a snail where they develop into larvae. From the snails, larvae end up in the water. But this time, larva lands on the water vegetation. You have to appreciate that it doesn't have to be like lake or river. It can be a meadow that is flooded once every so often. And then from the water vegetation, it can be consumed by cattle, sheep, or humans. Okay, and that's where adult worm develops. It goes from the intestine into the biliary ducts, and then it sheds the eggs. Eggs go back into human or cattle or sheep intestines and are passed into the environment. Does that make sense so far? So in this scheme of things, snails are the intermediate host, and cattle, sheep, or humans are the definitive host. Okay. Now, what are the symptoms of fasciolosis? Fascial Hepatica is also called a liver fluke. So obviously, obstruction of biliary ducts will lead to indigestion, fat malabsorption, fat intolerance, nausea. Microorganism. The worm can crawl all the way up to the liver, damage, therefore jaundice. But perhaps most intriguing is the role of this organism in the gallbladder cancer. Turns out that Fascial Hepatica feeds on the biliary epithelium, the epithelium in the biliary ducts and in the gallbladder. So this worm is essentially a farmer. This worm produces chemicals that stimulate the growth of gallbladder epithelium. Uncontrolled, because it feeds on it. It basically farms the epithelial cells. Controlled division of epithelial cells and accumulation of mutations in these epithelial cells will eventually lead to gallbladder cancer, basically a gallbladder carcinoma. This complication of liver fluke is very well known, and untreated fasciolosis almost ultimately leads to the gallbladder cancer. Does that make sense? Now I'm going to talk about a couple of nematodes, roundworms. The next one, Lumbricoidus, is the largest nematode that affects humans. Okay, so it all starts with the eggs in the environment. And they are consumed by humans. You know, fecal contamination of food products. Okay, hold on a second. I'm going to put human intestines. In the human intestines, eggs hatch into larva, and larva gets in the circulation, and circulation delivers it to the lungs. Okay, so let's talk about these steps. So when larva pierces through the walls of the intestine and ends up in the circulation, it takes some bacteria with it. So this bacteria that ends up in the circulatory system causes fever. And that's one of the first symptoms of Ascarious lumbricoidus infestation. Gram-negative bacteremia. That makes sense. When it ends up in the lungs, it irritates the lungs and people start coughing. And they cough larva up towards the larynx, where the mucus that contains larva is swallowed. So eventually, this larva end up back in the intestines. And here, in the intestines, larvae develop into an adult worm. Adult worm in the intestines starts shedding eggs. Now, you may in this overall situation, humans are both intermediate and definitive host. So humans are both. So first of all, I want to highlight that one worm is not enough to shed eggs, because these worms have sexes. So you have to have a male and a female in one gut to start producing eggs. However, once there are two of them, male and female, female can produce up to 100,000 eggs a day. Now, why these eggs end up on the human food? In underprivileged regions in the world, human feces are used as a fertilizer. So people collect night soil and put it on the garden, And eggs from that night soil might end up on the vegetation, and people then don't wash it properly and eat it, and voila, you have the infection. So by simply terminating this agricultural practice, you can break this cycle. What are the symptoms of Ascarious lumbricoidus infestation? Malnutrition, bloating, various mild digestive symptoms. The problem with ascariasis is that number of worms in the gut is limited by the size of the gut only. So there were cases of people having 2 to 3 pounds of live worms in the intestines. That leads to intestinal obstruction with vomiting and all that kind of stuff, and that can be treated only surgically. Okay, does that make sense? So basically what I'm saying is, if a person has a couple of worms and you give that person, say, mebendazole, the worm is dead and it is passed out with the feces. And you can see it and that's it. If you have 2 pounds of worms and all of them are dead, they stay. They just dead. They're just not producing eggs, but they're still dead and you still have to cut them out. Does that make sense? Okay. So the last nematode for today is going to be Strongyloides stercoralis. It's another roundworm. So eggs again. Well, let's start with the larva. There are different forms of larva, but we're going to just stick to the larva. So larvae from the environment can penetrate unbroken skin in humans, okay, and develop, eventually it will migrate from the skin to the intestine. So, adult worm in the intestine. The adult worm will shed eggs into the environment, and eggs will develop into a larva. Now, look at this. So first of all, humans here are definitive and intermediate hosts, just mentioning it. But look at this really interesting situation. Usually, the development, well, technically speaking, okay, humans can be an intermediate host. Technically speaking, humans are strictly definitive host in this cycle, and there is no intermediate, because eggs hatch into larvae in the soil. Does that make sense? And the worm can actually live in the soil for an extended period of time. Other human helminths that infect humans, they have to be in an animal. So intermediate host has to be some kind of animal, whether it's a snail or a sheep, as you remember, you know, or a pig or a cow, doesn't matter. Does that make sense? So, in this case, eggs can develop into larvae in the soil and exist in the soil. Now, when larva penetrates the skin, it starts to crawl under the skin looking for a place to enter the circulation. That leads to the formation of such symptoms as cutaneous larva migrans. Basically, it looks like a rash, but this rash changes. Does that make sense? Because those are tiny little larvae crawling under the skin and moving. They're moving fairly slowly, maybe a few centimeters an hour, but the pattern of a rash will change. Now, why I said human can be an intermediate host? Because adult worm can produce eggs that stay in the intestine, and they can develop into larvae in the intestine, and that may lead either to more worms in the intestine or to the systemic infestation. Does that make sense? So here you can see that humans serve not only as the definitive, but also as the intermediate host. This part right here, okay, this is called a superinfection. This type of infection with Strongyloides stercoralis is quite rare in immunocompetent individuals, but in people who are immunocompromised due to the malnutrition or immunosuppressive therapy, some kind of steroid therapy, that may lead to superinfection. Now, what are your chances to encounter that kind of a patient? They're pretty high because Strongyloides stercoralis is pretty common in Central and in Latin America. So you may have patients who recently emigrated from those countries that may have chronic strongyloidiasis. It's not unheard of. Does that make sense? Any questions? I'm going to stop recording.